Hello everyone, my name is Almosawi.
I'm Catherine Len.
I'm Justin Darragh.
And I am James Gaecke.
And we will be presenting a review about an article called Genetic and Environmental Causes of Variation in Epigenetic Aging Across the Lifespan by et al. So what is epigenetics? Epigenetics is an alteration to the DNA structure that affects gene expression. Essentially, it is the changes in the chromatin structure. What is epigenetic aging and how is it measured? Epigenetic aging is the biological clock of an organism, and it can be used to estimate the actual chronological age. Typically, this is determined by measuring the level of DNA methylation. So then, what is DNA methylation? DNA methylation is the addition of the methyl group to the nucleotide bases. So as you can see in the picture, in eukaryotes, the hydrogen in the cytosine group is replaced with a methyl group, which produces 5-methyl-cytosine. So methylation alters the activity of a gene, more notably when a promoter region on a gene is methylated, transcription is suppressed. So then how is transcription suppressed? The presence of a methyl group in the major groove of DNA inhibits the binding of transcription factors and other proteins that are required for transcription. So this study will utilize DNA methylation biological age, known as DNMH, which is a predicted age based on the DNA methylation levels that are measured from a DNA-based source. This is a vital biomarker for adult health. What can DNAMH tell us? DNAMH reports lifestyle factors, disease risk factors, and genetic variations. So then why is this study in a research article? While methylation can be used to make a rough estimate for aging, loss of methylation can also predict future health problems and risk of disease. DNA methylation can vary greatly due to environmental factors. So since methylation is associated with health, it could be crucial for future medicine to parse out what environmental factors cause loss of methylation. The article concludes that the variations are almost entirely due to environmental factors. So the importance of the study, it is the first study to provide a comprehensive view of the genetic and environmental causes of DNAMH variation across the lifespan. Next.
So on to the study design. This study is considered a meta-analysis, and a meta-analysis is just a statistical analysis that combines the results from different studies that address the same question or focused on the same topic. DNA methylation data was pulled from 10 studies that had 4,217 participating individuals that ranged in age from 0 to 92 years old, and they all came from 1,871 families. These studies were accessed through a public repository and primarily came from Australia and Denmark. And most of these studies measured methylation using DNA extracted from peripheral blood and utilized the human methylation 450 array. One study used an EPIC microarray using cord blood, and 4 studies used the 27K array, which utilized cord blood, human umbilical vascular endothelial cells, or the placenta, as the biological sample. However, most studies use the peripheral blood and the 450 microarray. So what is a 450 array? This is just when they analyze over 450,000 sites where cytosine is next to guanine in the DNA sequence. Analyzing data. So the Horvath epigenetic clock was used to determine DNAM age, and this tool is used primarily because it was developed to work across tissues and ages. And the 353 methylation sites used by this clock are common to the 3 methylation arrays used by the 10 studies where the data was collected. So a Horvath epigenetic clock bases their age estimate off the 353 epigenetic markers on the DNA. And then these markers measure DNA methylation of CpG dinucleotides. The Horvath clock is special because it allows users to compare and contrast the ages of different tissues from the same subject. This means that it can be used to pick out which tissues, if any, show signs of accelerated age from disease. The 2 other clocks that were used to corroborate what the Horvath clock estimated, they were used because they were also applicable to the data and to corroborate what the Horvath clock estimates. To account for the effect of chronological age on DNAM age, researchers studied epigenetic age acceleration. This value was calculated as the residuals from a linear regression of DNAM age on chronological age. In addition to this, sensitivity analyses were performed for studies that measured DNA methylation in blood to ensure that the results in the cell composition were adequate. And then the different pairs associated with the multivariable normal model and the Fisher program were monozygotic twins, dizygotic twins, siblings, and spouses. A likelihood ratio test was also used to compare nested models, and a likelihood ratio test assesses the goodness of fit of two competing statistical models based on the ratio of their likelihoods. All p-values in the study were two-sided, and any p-value less than 0.05 was considered to be significant. The familial correlation across lifespan was modeled as a function of cohabitation among the pairs.
All right, so results. Researchers found there to be a moderate to strong correlation between methylation and age, as expected. Results for monozygotic and dizygotic twins showed that methylation correlated from a very young age even up to old age. This shows that researchers already knew for the most part that methylation has genetic factors and that it increases with age. Next slide, please. DNA methylation can predict actual chronological age within about plus or 15 minus years. An expected margin of error for a test involving multiple kinds of tissues like this. Next slide, please. Within families, researchers found that methylation correlation for twins actually started as very low at birth, but continued to increase sharply with age. Correlation for spouses and parent-offspring pairs also increases with time. Once twins separated and began living apart, correlations plateaued a bit. So you can see at that point, it's the point where it stops correlating and starts tapering down a little bit. But that correlation still persists into adulthood, although it doesn't increase. Genetic factors were estimated to only explain 13% of the variation. So what does all this mean and why is it important? This shows a significant amount of the DNA methylation in humans occurs because of environmental factors and not just because of genetic factors. DNA methylation seems to also be very dependent upon the conditions during childhood, as you can see with the monozygotic and dizygotic pairs. Even the dizygotic has very strong correlation. They start near zero, but as you can see, extremely sharply increase with time. This highlights the importance of determining what environmental factors affect DNA methylation in humans. Since DNA methylation is a strong predictor of health and disease in the later stages of life, and since DNA methylation is determined heavily by factors during childhood, identifying environmental risk factors for children could potentially play a very important role in preventing disease for individuals in middle to old age. While this study proves that environmental factors play a key role, more rigorous research is required to identify the specific risk factors. Next slide.
Okay, so our opinion. Among many of the implications discussed through this study, the effect of cohabitation, normalizing DNA M variation among groups of individuals that cohabitate or live together, is a very strong indicator of how environmental factors can affect DNA M age. So much so that, as kind of stated in the results, individuals that live together have almost like a normalization of the rate of variation, and then when they grow apart, it changes. Environmental factors are discussed a lot throughout this paper, and noted that it's a very broad topic. There are many different types of environmental factors and inputs, both from physical exposures such as chemical or radiological to psychosocial exposures such as stress or anxiety, having an effect on variation. We consider this meta-analysis to be quite reliable. The sample size is large, the statistical analysis by the authors appears sound, and the author is able to perform an inclusive examination of different correlations by performing an analysis of the whole lifespan. They also note that it was one of the first studies to do so, with previous studies focusing more so on the different stages of the lifespan. The authors did make a note, and it was discussed in results, that when examining environmental inputs, genetic input only had around 13% only changed variation by around 13%, with genetic factors accounting for that, with a majority of change in variation occurring from environmental inputs. Next slide. So looking forward at future research that could be conducted, DNA M age and chronological age had some degree of variation between each other. And we did note that there were different sites of tissue sample collection to measure the DNA M age. Future studies might seek to study the DNA M age variation among single types of tissue samples. So we noted that there was placental, endothelial, peripheral blood, and vascular tissue collection that were used for the different samples. Of course, this being a meta-analysis, perhaps future experimental studies could look at rates of variation among a single type of tissue. One strength of the study was the decision to examine variation over the course of the entire lifespan. We were wondering if future studies may look to determine if the rate at which variation develops is accelerated over different types of environmental input at different points during the lifespan. We did note that the author stated that the variation was largely set before adulthood, but perhaps there are different rates at which it occurred throughout the lifespan with different types of inputs. While environmental factors are a broad category, future work may look to examine how the deleterious changes in DNA M age variation can be rectified or prevented due to its correlation with disease risk. And can preventative medicine be helpful in preventing this? As we noted that there was a large correlation with disease risk. What can individuals in the preventative medicine field do to ensure that any of these deleterious changes can be subverted? Are there environmental factors that affect disease risk at different rates? Do certain environmental factors change variation faster? Do certain environmental factors affect it more slowly over different types of time? We also wondered if certain individuals in certain geographic locations or different demographic data were more at risk to some of the more deleterious or harmful DNA M age variation changes simply due to the environment that they live in, and what can be rectified to kind of subvert these changes. Next slide.
And these are just our references. We just used the article that we found and the textbook.